Welcome to Bachelor, where we help microbes find their perfect match. I'm your host, Eva, and today we have a mysterious new profile that's catching everyone's attention. Let's go look at their profiles. Okay, let's look at Golden Staff first. For their bio, they said they're just a friendly cluster-forming caucus looking for a warm and welcoming host cell. They love hanging out on skin and getting cozy in wounds. They're pro at breaking down tissue. I'll definitely break down all your walls, is what they said. Their interests are protein A production, biofilm formation, spreading to new territories, and they're looking for an open-minded epithelial cell for initial attachment. Okay, let's look at Epi the Guardian. Their bio is holding down the fort at the skin barrier, looking for someone who respects boundaries. Their turn-ons are good barrier function and tight junctions, and their red flags are suspicious surface proteins and toxin production.
When I saw that epithelial cells profile, I just knew we'd stick together. My adhesions were tingling.
He seems nice, a bit clingy maybe, but who doesn't love a strong attachment?
Let's talk about how our bacterial bachelor makes the first connection.
S. aureus uses several clever strategies to attach to host cells and avoid detection by the immune system, and these strategies are facilitated by proteins known as adhesions. One such adhesion is protein A. Think of it like a bacteria's fake designer outfit. It looks fancy, but it's actually just to help it avoid being recognized as dangerous. Basically, it does this by bonding to antibodies like IgG, especially in the Fc region, and prevents the immune system from recognizing the bacteria as a threat. It basically interferes with the normal immune system, allowing the bacteria to go unnoticed by immune cells. Next, we have the clumping factor, which acts like a super strong relationship glue. The adhesion binds to the fibronectin and other proteins found in human tissues and bloods, and it basically helps S. aureus attach firmly to surfaces like skin, mucous membranes, and blood vessel linings. It locks the bacteria in place, allowing it to persist in these areas and potentially cause infection. Lastly, we have the fibronectin binding proteins, and these function like the bacteria's hands, reaching out to grab onto specific surface proteins on the host cells, particularly fibronectin. This interaction helps S. aureus anchor itself to tissues and cells, kind of like a clingy date that refuses to let go. These proteins enable the bacteria to stay attached and form biofilms, which protect it from both the immune system and antibiotics. All these adhesions work in tandem to ensure that S. aureus sticks around and maintains its presence in the host.
But this seemingly perfect match is about to turn toxic. Literally.
Hey baby, want to see my protein A?
Is that toxins in your pocket?
So PAMs were just detected. Now we're really seeing the true nature of S. aureus' toxic personality.
S. aureus has a dangerous set of virulence factors that help it invade and evade the immune system. The A toxin creates holes in the host cell, the coagulase forms protective blood clots like a fortress, and the enzymes break down tissues to allow the bacteria to spread. The capsule acts like a shield, hiding the bacteria from immune detection, while the biofilm forms a protective barrier that blocks antibiotics. Together, these factors are the red flags that the immune system should recognize in order to fight the infection.
Security, we've got a cold golden. Release the cytokines.
Backup needed! Suspect armed with virulence factors!
Here comes security, the innate immune response. It's like the club bouncers of your body. Let's talk a little bit about it. The innate immune system is the first line of defense against Staph aureus or any other pathogen and provides a rapid and non-specific response. Here's how it works. The first step is detection. Epithelial cells such as skin and mucosa act as physical barriers. If breached, they release antimicrobial peptides, AMPs, like defensins and calphallocidins, which will kill the S. aureus. Pattern recognition happens when immune resident cells detect S. aureus via pattern recognition receptors like toll-like receptors and nod-like receptors. The second step is early responders, which are macrophages, which were the security guards. The tissue-resident macrophages will engulf S. aureus through phagocytosis. They'll secrete pro-inflammatory cytokines such as IL-1 beta and chemokines such as IL-8 to call for backup. They will also process antigens to show the adaptive immune system later. Mast cells also release a histamine and other mediators that will increase blood vessel permeability, helping immune cells and access the infection site. The third step is the recruitment of neutrophils, which will arrive in large numbers via chemotatic signals, such as IL-8. They'll perform phagocytosis and release toxic molecules such as ROS and lysosomal enzymes and NETs. The next step would be amplification, as the complement system enhances phagocytosis and triggers inflammation, as well as recruiting immune cells, while pro-inflammatory cytokines will amplify local and systematic inflammatory responses to help combat S. aureus. This case of bacterial deception has been elevated to the highest court in the immune system.
Will the defendant please explain the toxins that we found?
Your Honor, we're producing antibodies as we speak.
Order in the court!
You just saw us in the high security court system, where the adaptive immune response was being triggered. Let's talk about it.
Dendritic cells capture pathogens like S. aureus and present their antigens to T cells and lymph nodes, ensuring that the immune system targets the correct pathogen. T cells, which includes helper T cells and killer T cells, interpret this evidence and coordinate the immune response. Helper T cells activate B cells, recruit other immune cells, and guide the response, while killer T cells destroy infected cells by inducing apoptosis. B cells produce antibodies tailored to S. aureus with IgG neutralizing toxins, IgA protecting mucosal surfaces, and IgM acting as a first responder. The adaptive immune system's specificity and memory allow for a faster, stronger response to future S. aureus infections, reducing the likelihood of reinfection.
And that's how our bacterial bachelor, S. aureus, got banned from the dating app. But don't worry, our immune system created a block list that will last a lifetime. And lastly, we're going to talk a little bit about the immunological memory. Just like how dating apps remember your swipe history, your immune system remembers memory cells.
Imagine S. aureus, a bacterium known for causing infections, tries to match with your immune system. On the first encounter, your immune system takes its time to study and respond, just like swiping through profiles on a dating app. But your immune system doesn't forget it. It saves this profile for future references by creating memory cells. These cells make sure your immune system is ready to act if the bacterium ever tries to invade again. First, memory B cells are like detectives. They store the precise molecular structure of S. aureus' antigens, specific proteins on its surface. When reinfection occurs, they rapidly produce large quantities of highly specific antibodies to neutralize the pathogen. Memory T cells are the warriors, and they have two primary roles. Cytotoxic T cells remember how to directly attack infected cells by releasing toxic molecules. Helper T cells coordinate the immune response by signaling other cells, boosting antibody production, and activating other immune system components. These memory cells can live for years, even decades, which circulate in your body and remain on high alert. During the first infection, your body activates naive B and T cells. This process can take between 1 to 2 weeks to mount a full defense because your immune system is essentially learning how to fight the invader. With memory cells, if S. aureus strikes again, your immune system responds much faster. Within days, memory B cells release specific antibodies and memory T cells target infected cells directly. This secondary response is not only faster but also much stronger. Memory cells produce significantly more antibodies, which are highly tailored to S. aureus compared to the first response.
Thank you for tuning into Bacterial Bachelor.